The core element of my research concerns itself with the mechanisms of learning and memory, how we acquire new information about the world, and how we retain it in memory storage over time. And recently I've become interested in disorders of memory, how memory storage can go awry in various disease states, either in the aged or in the very young. It's important to know how we learn and remember. It's important for schooling of our children, for the education of society, to get some insight into the biological basis of memory storage. But in addition, the biology of memory is key to understanding how memory goes awry, how it becomes distorted, how it becomes lost, what happens in Alzheimer's disease, what happens in age-related memory loss, why are some children born with mental retardation, why are there cognitive deficits in schizophrenia. All of these questions are based upon the nature of learning, and particularly in the nature of memory storage. And the biology of memory storage is likely to provide the foundation for addressing these disorders of cognition. I've worked on two experimental systems in order to look at the molecular mechanisms of memory storage. The first is I worked on a very simple snail, the marine snail Aplysia, which has a very limited number of cells, 20,000 nerve cells, and so one can work out the neural architecture of a behavior in extraordinary detail. And there we were able to look at very simple forms of learning and show that they involve strengthening of connections between nerve cells, and that long-term memory differs from short-term memory in two ways. One, it involves an alteration in gene expression, and two, it leads to anatomical changes in the connections between cells. Short-term memory involves a functional change, long-term memory involves an anatomical change. But in the work on the snail, one was limited to very simple forms of learning and memory. One also wanted to explore more complex forms of learning, the kind that involve thinking, planning, strategy, and part of human beings. And that I realized one could model very effectively in the mouse. And in 1990, when I realized one can use genetically modified mice, in which one can alter one gene at a time, we began, my colleagues and I, to use genetically modified mice in order to explore more complex forms of memory storage. And we found that although the details differ significantly, the basic principles are the same. Learning involves a strengthening of synaptic connections, and long-term memory differs from short-term memory in involving a growth of anatomical connections between cells, and that a genetic switch is necessary to convert short-term to long-term memory storage. Well, I was really struck with what an inspiring opportunity working in a laboratory was for me, and how it gave me a completely different view of the practice of science that one gets from taking an organic chemistry course or a course in Introduction to Physics. And what I do is I encourage high school and college students to come and spend a summer in my lab to see what it's like in a practical sense, because the practice of it, the thinking about the problem, the doing the problem, the social interactions of a lab are so exhilarating. The opportunity of finding something new, even if it's a small addition to knowledge, is fantastically inspiring and energizing. So I think working in an active lab is the best advice I can give to a young person.